So what we're looking at is the structure of the DNA here, and in 1953 Watson and Crick were the ones that established the double helix model of DNA in there. And when they were searching for it, the Morgan's research group showed that genes were part of chromosomes, but they didn't know what exactly the genes were made of. And at that point, the DNA and the protein became the candidates as to what they thought this genetic material was. So they weren't really sure. They knew there was these genes that were inherited, but we didn't know physically what they were in there. So the role of DNA in hereditary was worked out studying bacteria in there, and bacterial transformation was the key to understanding it. Transformation is a change in the genotype and phenotype due to taking up external DNA. So they used bacteria in there that took up DNA from the outside environment and therefore changed its genotype and phenotype to establish that that DNA was what was actually carrying the genetic information in there. So bacteriophages or phages are viruses that will infect bacteria in there. And a virus is basically a piece of DNA or RNA that's enclosed by a protein coat. It doesn't have any organelles or anything like that. What it has to do is go into the host cell and use the host cell's machinery to create new viruses in there. So viruses are not really considered to be alive because they can't do things on their own. It's mandatory that they go in there, a mandatory parasite. So what they studied was the T2 phage that infected E. coli in there. With these phages, they were named T and then different numbers in there in the order that they were discovered. And they just happened to use T2 in here. And they used these radioactive proteins and found that the phage DNA entered the cells and the radioactive proteins remained outside the cell. So they found out that it was the DNA that went in, not the protein in there, and the cells released new phages showing the DNA that functioned as the genetic material of the phage of T2. So there was additional evidence that the DNA is the genetic material. Erwin Chargaff, he studied the ratiotides of the nucleotide bases, which are the A, T, G, and C, and he found that the number of A's is approximately equal to the number of T's in any cell's DNA, and the number of C's is approximately equal to the number of G's. So the reason for that is because C and G pair together. If you have a C, you need to have an equal number of G's to go with them. Same thing with the A's and the T's in there. So they came up with Chargaff's rules, which is the base composition varies between species. So the percent of A's and T's and G's and C's that any species has will vary. It will be similar for all the organisms in the species, but say between a cat and a dog, it's going to vary in there. And within the species, the number of A and T bases are equal and the number of G and C bases are equal. So when we build the DNA model, it's going to be in the shape of a double helix. So it looks like a ladder where you've got the two sides of the ladder and then it's twisted in there with the rungs of the ladder in between. And it's arranged anti-parallel, so the two subunits of the sugar phosphate backbones are going to go in opposite directions. So you've got that 5' to 3'. It's going to go, say, 5' to 3', and then 5' to 3' will line up in the opposite direction over it. So it will be like this in there. So your nitrogenous bases get paired in a specific combination. You're going to have one purine and one pyrimidine in there. So when you have A and T paired together, one's a purine, one's a pyrimidine in there. So your purines are C, T, and U, and your pyrimidines are A and G. So A and T pair together, and they're held together with 2 hydrogen bonds. G and C pair together, and they're held together with 3 hydrogen bonds in there. So this is just showing that you've got this double helix. This is showing where you've got your purine and pyrimidines in here, 5' to 3', 5' to 3'. And then this is more of a molecular model form of it. So when we make DNA, we use a semi-conservative model. So what it means is you're going to take your parent molecule and you split it in half, and then you're going to fill in the other half with each parent in there. So it's like if you start with mom and dad, they get divorced and married new people, now you got two couples in there. So, yeah.
When you look at the diagram, why does it go ATCG, TAAT instead of going like back and forth? Like ATCG, TAGC.
So A always has to pair with T, and G always has to pair with C. If you look at it, you'll see this is kind of a pointy shape, and this is kind of a round.
Well, I was asking, why is it in the order that it's in?
Oh, they just randomly make it up on here. But the order will determine what proteins it codes for. But on the models like this, it's just randomly created. So what you end up with is half of the original, half of new material for each of the daughter strands that are made in there. So when we go to do this replication in here, you're going to have an origin of replication. So on here you can see you've got this, they like to use the bacterial one as an example for this because their DNA is going to be circular. And so you can see that you've got this origin of replication where you start to pull the two strands apart. So it's going to create a bubble in there. And then the bubble gets bigger, and when you do that, you create two replication forks. So you can see there's a fork in the road here and a fork in the road here. It's able to copy it in both directions in there and go towards the other end. When you do this with eukaryotic cells in there, you can have multiple origins or replications, and then you end up with multiple forks in there. And so you just keep pulling the bubbles apart until the bubbles fuse together and it becomes separated in there. So the replication fork is just that Y-shaped region at the end of the replication bubble. So when you do that, if you take like yarn or some sort of rope and you go to pull it apart, you'll notice that it coils up tighter on the other ends of where you're separating it in the middle. We don't want that to happen with the DNA, so we've got these helicase enzymes that are going to help to untwist it ahead of time so that it can pull it open easier in there. And then to help keep it open, we've got the single strand binding proteins in there that will pair to it in there and help keep it open. And the topoisomerases are going to help to relieve the strain ahead of the replication fork in there by breaking and rejoining the replication strands. So when you're untwisting something like that, if you're trying to untwist it ahead, at some point you're going to have to break it open, untwist it, and reattach it in there. Otherwise, that knot that you make just keeps getting moved farther and farther out. So that's what those are going to help to do. So the enzymes that synthesize DNA cannot initiate the process. They can only add to nucleotides at the end of an existing chain, and they have to be base paired with a template strand. So there has to be these little sequences in there to get things started. And that's where we have the primer come in. And the primer is the RNA chain of initial nucleotides that will be produced during DNA synthesis. So that primer is made of RNA, which is a little bit different than the DNA in there. And you've got an enzyme that's going to help to control that, the primase that's going to synthesize the primer. And it starts a complementary RNA chain from a single RNA strand on there. And then it will add the one RNA nucleotide at a time. Primers are usually 5 to 10 nucleotides long, and the DNA starts at the 3' end of the primer. So what you have is So you're going to have your section of primer that's 5' to 3'. So this is your primer, it's RNA, and then you have your DNA here. So it continues, the DNA starts at this 3' end of the primer in there. So it can be a little confusing when you've got 5', these prime ends and primers and primerase in there, because it all sounds alike. But you guys got those to work pretty quick today. Once you have the bubble in there, there's not a whole, you see what's going to happen in the lab. Well, I guess you can wait and see what happens if it gets bigger after a couple hours or if it blows it off. It's the closest we're going to get to having an explosion in the lab. Woo! You popped your balloon off.
Balls, let me have use of your rooms and I can have it.
Well, we could all make an explosion in here, but I'm not supposed to have an explosion. So when we synthesize the new strand of DNA, then you're going to use a DNA polymerase. So when we synthesize the new strand of DNA, then you're going to use a DNA polymerase. And a polymerase is just going to make a polymer out of something.
A balloon is erect.
Yes, it is. And so with eukaryotes, there's at least 11 different polymerases. So with the eukaryotes, there's at least 11 different DNA polymerases. In here, the only thing I'm going to expect you to know the difference is when you use a DNA polymerase and an RNA polymerase. I'm not going to ask you to know which DNA polymerase in there. So DNA polymerases are going to require a primer and a DNA template strand in there. It's going to work on DNA, and RNA polymerase works on RNA. The DNA polymerases will add nucleotides to the primer at about a rate of 500 nucleotides per second in bacteria, and it will add on about 50 per second in human cells. So it's a little bit slower in human cells, but bacteria can also replicate and double pretty quickly in there. So each nucleotide added is going to come from a nucleoside triphosphate, and then the monomer joins the growing end of the two phosphates that are lost in the coupled exergonic reaction. So the phosphates coming off when you add it on there, it's going to provide its own energy to add it in there. So they're going to have anti-parallel elongation in here. So what that means is you've got these strands that run 5' to 3'. If it's going to always add on to the 3' end in there, then this one is going to elongate in this direction. This one elongates in this direction. So they have to elongate in separate directions. And you can't do that very practically when it's opening up and splitting in this direction. So what we end up with is a leading strand that's going to require only one DNA polymerase, and you'll have a lagging strand that's going to need multiple DNA polymerases. So if you have this opened up, you have 5' to 3'. When you add on to this one here, it just keeps adding on as you open up, going down this direction. This one is, or I'm sorry, it's going to keep opening up, going this direction in there as you open. Why am I? You know what? I'm going to use the picture that's in here. That's going to be better in there. What's going to happen is one has to go like this. The other one has to go this way. And it has to do it in small segments. So it's going to come and go here, come and go here, come and go here, come and go back this direction. And that'll make more sense with the other picture in a minute in here. So your lagging strand, it's going to make these little fragments that are called Okazaki fragments in here. And they're going to have to be joined together. And you're going to use a DNA ligase that are going to join those together. And those are about 1,000 to 2,000 nucleotides long. In E. coli, in eukaryotes, they're 100 to 200 nucleotides long. So this is showing it here where you've got the leading strand, and then you've got the lagging strand coming along behind from the replication fork in the replication bubble. There. So does that make sense what we mean by a leading strand and a lagging strand? Because it always has to go 5' to 3' in there. In one direction you get to go continuously, but in the other direction you have to keep jumping out and going back like this. So when you have the different proteins, the polymerases and the DNA in there that are all hooked together, this is going to be your DNA replication complex. So in reading this, you can read that pretty fast. If you're reading in there at 500 nucleotides per second and adding those on, that's like trying to imagine typing 500 keystrokes per second in there. That's pretty fast in there to be reading this. So some mistakes are going to happen in there. And we've got proofreading mechanisms that are built into this to help catch some of those mistakes. It doesn't catch all of them, but it does catch a good portion of them. So your errors in your completed DNA only have about 1 in 10 to the 10th errors in there that are just mistakes that are left, that are missed. Your initial pairing has 1 in 10 to the 5th nucleotides in there. So you reduce the errors by half in there. And so during replication, the DNA polymerase tends to proofread itself in there, but sometimes it's going to miss things in there. So some of the types of mistakes you can have, the mismatch repair in there will go in, and you'll have other enzymes that will look at that and say, okay, we had the wrong DNA in here. It's going to cut it out and replace the new one in there to fix the errors in there. So DNA is going to constantly be exposed to mutagens, and the errors a lot of times are corrected, but sometimes they're not, and then you can have permanent damage. So mutagens are just substances that will cause DNA mutations. A lot of them end up being carcinogens in there because if you get enough mutations, it will lead to cancer, and a carcinogen is just a substance known to cause cancer. In the E. coli, they've identified almost 100 repair mechanisms for correcting mistakes in DNA. In humans, we've identified about 130 correction mechanisms in there. So we do have a lot of ways of fixing those mistakes that are in our DNA. And that's why you can be exposed to mutagens quite a bit, actually, and not have cancer absolutely every time in there. Is our body does have means to fix it. It's just how often do you want to invite the errors in there and hoping that it gets fixed. And statistically, if you expose yourself to enough errors in there from the mutagen, you will likely end up with it leading to a problem like a carcinogen. So a nuclease is a DNA cutting enzyme which is going to cut out the damaged DNA, and it is going to have a gap that gets filled in with nucleotides. So we call that nucleotide excision repair, where the enzymes involved filling in the gap come in. So one type of mutation that we can have is a Thymine Dimer, where the thymines next to each other become covalently linked, and then they're no longer read as thymine anymore. Anybody have any idea what might cause thymine dimers that we're all exposed to on a daily basis? This substance also causes a type of cancer. No, because you can choose not to have chicken. UV light. So UV light can cause the thymine dimers in there. We fortunately do repair most of them, but it's another one of those things where UV light, you can't live without it. If you have no UV light, you will not survive.
Why is that?
I think the damage from the UV light, a lot of it was filtered more so by the ozone layer in the past and is not as filtered now, but there's also a lot more other things in there that can add to the mutation rate in there. One of the things they found was, for a long time it was such a push, everybody have sunscreen, everybody wear sunscreen. Well, they found that some of those sunscreens are actually carcinogens. It's not the physical barrier, but the chemical sunscreens that are more carcinogenic in there. The ones that are like the titanium oxide and stuff, that's not carcinogenic because it's just a physical barrier on your skin. You need it for vitamin D in there. You will not be healthy. So like a lot of times when people have like lizards and reptiles as pets, those normally live outside in the sun in the desert. And sometimes people get them and they won't put a UV light bulb in there. They'll think, oh, I'm just going to use a cheapo fluorescent light bulb in there and give them a warm stone to sit on. And their lizards will get really unhealthy. They'll have bone disease in there. And we know that a lot of cancers are associated with decreased vitamin D too. So. I don't feel so bad for them though. I think they do get to go outside though. Yeah, with some of them I don't. I think if you're like serial killer. No, but it's not that they're a healthy individual to start out with, but yeah, it's other health effects. I mean, I don't think you would necessarily, it's not like you're going to say, oh, I've had no sunlight for 3 weeks, I'm going to die. If that was the case, we'd all be hosed up here. But it's just your better health with it in there. So the alterations in DNA are significant for evolution because sometimes some of those mistakes will slip through and it will introduce something new into the gene pool that's actually better. Sometimes it's worse, sometimes it functions the same in there, but it does introduce more variation into the gene pool. So what we have though when we are replicating DNA is it goes 5' to 3' like that, but you can only add to the 3' end. There's a small little section here that there is no making a 3' out of that. It is the 5' sugar on there. And what happens is this doesn't get replicated or repaired. So next time it's going to be a little bit shorter. And the next time, a little bit shorter. So what will happen is that DNA is going to progressively shorten in there. So if you have a circular chromosome, that's not a big deal because it's all continuous, but in eukaryotic cells, we don't have the circular chromosome. So that we don't end up with our DNA shrinking off to nothing, what we have on the end are these segments that are called telomeres on there. That would be kind of this additional segment on here, so that you can shorten the telomere instead of the DNA in there. So the telomeres in there are going to be a short 6-nucleotide sequence in there, but you have multiple telomeres on there. So each human telomere is going to have a repeat of TTAGGG that's going to be between 100 and 1,000 times on there. That's kind of your buffer zone for getting chopped off in there. And so it prevents staggered DNA that leads to cell cycle or arrest or cell death in there. So telomeres are one of the things that are associated with aging, is that they would shorten over time in there. And so there's been some kind of interesting studies done out there looking at things that cause you to have your telomeres shorten and not shorten. And they've looked at it where I look up all kinds of research all the time with running and endurance sports in there, just out of my own personal interest, hoping that one day I'm going to make myself faster, that I don't have to be at the end of the group all the time. Yet to find the magic thing that helps that. But what I did come across was some research, though, with the telomeres, and that they thought that with all of this extra activity, increased metabolism in there, that it would cause a person to age faster in there and shortening the telomeres. And what they found is that they actually don't shorten in there, that there is something that goes on and stops them from shortening in an endurance event like that. So there are mechanisms that will stop telomere shortening temporarily for us. So a telomerase is an enzyme that is going to catalyze lengthening of the telomeres in a eukaryotic gene and restores their length. And the telomerases are not active in most human somatic cells under most conditions in there. It's thought that the telomere shortening in there may actually help protect us against some cancers, because when some cancers replicate, if they shorten all the telomeres, then all of a sudden you can't shorten them anymore and you're done replicating. It would end their life cycle in there. However, a lot of cancers actually have telomerase activity in there as a way to bypass that. So there are some cell cultures that they've used, and what they used to find was that you could get these cell cultures, but they would not be able to live for very long. A lot of cell cultures would be good for about 50 cell cycles, and then they would die with eukaryotic cells. And by having cell cultures that were derived from tumor cells in there that had the telomerase activity, they were actually able to get these cell cultures that didn't die. And so one of the more common ones are the HeLa cells in there, which there's a little bit of a section, and we'll talk about that a little later in there. So when we make the chromosome in there, the chromosome is going to consist of the DNA molecule that is packed together with the proteins. The nucleoid is going to be the region of the DNA in the bacteria. They don't have a nucleus, so they just cluster it together in one region. The chromatin is going to be your complex of DNA and the associated proteins in there. It's in an uncoiled state is the chromatin. In order to get the DNA to coil up into chromosomes, we have to have these histones in there that are small little proteins that more than 1 in 5 are positively charged with lysine or arginine to bind tightly to the negatively charged DNA in there. And what these are going to do is allow the DNA to wrap around them in there. So it's the first level of packaging. The four most common types are H2A, H2B, H3, and H4 in there, but there are other types. So what's going to happen is you're going to have these histones that are going to have the DNA wrap around each histone twice, and that is going to create a nucleosome. So that bead of DNA around the histone. And then the DNA between the beads is the linker DNA. So these histone tails of one nucleosome and the linker DNA are going to interact to make a coil that is a 30 nanometer fiber. So those coil up like this. So it would be kind of like saying you take your hair and you make, I don't know, 30 different braids. And then you take 3 of each of those braids and braid those into another braid. And then you take those other braids and braid those into another braid. You've got levels of coiling folded on top of each other here to do that. So we have the loop domains, which are loops of the 30 nanometer fibers that will be attached to the chromosome and scaffolded in there to make a 300 nanometer fiber in there. And eventually these loop domains coil and fold to make your metaphase chromosome in there. And it's going to further compact the chromatin to make a chromosome with a width of 700 nanometers in there. So all of this folding, while it looks like it would be random in there, it's very precise. You can take the strand of DNA and it's always going to fold up the same way around the histones, making the nucleosomes and the loop domains, so that you end up with the gene in the same place on the chromosome every time it folds. So heterochromatin is the interphase chromatin. This is visible as irregular clumps, and this is the unfolded state. By being unfolded in there, enzymes are going to be able to get in and access the DNA to be able to use it. The euchromatin is less compact, it's more dispersed in there, and it's going to have looser packing, making it good for transcription in there. So that's the end of